welcome back from our short break. So I would like to welcome our next speaker, Dr. Karl Morten from University of Oxford, presenting the talk titled Energetic Dysfunction in ME/CFS and Potential Relevance to Long COVID.
Thank you. Can you see my slides okay? Are they there?
Yes.
Yeah, okay, great. So thank you very much for the organisers for giving me the opportunity to talk today. So actually, this is the only talk on the programme that's not about long COVID, so it's a bit of a challenge. So a bit of background on me. So I've worked on mitochondria or biology for the last 30 years. So I'm based in Oxford at the Women's Centre at the JR. And the two people on this side that are responsible for me getting involved in ME/CFS, there's the late Norman Booth, who is a physics professor in Oxford, whose wife has ME/CFS, and Charles Shepherd, who's basically funded all of our research over the last 3 or 4 years. So they've dragged me into this. It's been a challenge, but a very interesting one too. So I'm going to take you through some of the work that we do. So just to set the context really. So this is a slide just showing how do we understand these chronic conditions. There's very many chronic conditions out there. So we don't know what the cause is necessarily most of the time. There's lots of comorbidities, which we've heard about this morning and yesterday. And there's a trigger at some point that kick these things off. And there's probably a driver that is driving the system now. And it's trying to understand how this all fits together. So in terms of long COVID, we know about COVID-19, which was the cause potentially. Or maybe it wasn't the cause, or maybe it's just part of the process. It was interesting in hearing Melanie's talk in the earlier about that half the patients that had POTS had something like that before, so they were vulnerable. So there's clearly some susceptibility factors in long COVID, and probably in other conditions as well. So there's a driver that's driving the disease now, and obviously that's the key factor for us in trying to work out what we could do therapeutically to sort of slow this down or even treat the condition. In terms of ME/CFS, we know a lot more about that. We've been working on it for quite a long time. So the panel on the right just shows you some of the symptoms that we find commonly associated with the condition that we pulled out of a UK ME/CFS biobank. And you can see on the right-hand side, there's different severity groups that we've looked at. There's mild patients who probably could have normal jobs. Moderate ones may be housebound, but they can get out and about a little bit. And then there are really severe patients that are bedbound all the time, pretty much. And you can see as you go across on the heat map, as you get more severe, the symptoms become more encompassing. And what we found over the last few years is really how important it is to have other groups that are also fatigued. So we've been looking at a multiple sclerosis cohort, and you can see they're very similar in a way to the mild patients in terms of the symptoms are very similar, and obviously a healthy control. But in terms of what drives ME/CFS, we really don't know. Many patients talk about a viral trigger, possibly EBV from glandular fever, or maybe a gut infection, or even stress can be a trigger. But there's clearly other things that these people are vulnerable, other factors that are associated with them that are part of the process in why they are vulnerable to developing this condition. Because not everybody that has EBV or other things gets ME/CFS. The disease driver is key, and this is something we really try to see whether we can find out. This is an interesting slide. It shows you some of the problems with this condition. It's quite an invisible condition. There are people, as you can see on the top right, this is a strong family. So Jamie in the middle works with me. He's quite seriously infected, but he can actually work. And Laura, his sister, also has ME/CFS, and they look completely healthy when you see them like this. But Laura is housebound. She can't really get out and about. And Jennifer Bray on the top, who produced a really superb documentary called Unrest, also looks really healthy. And then you've got the other extreme, where Whitney Defoe, whose father is quite a famous scientist called Ron Davis, is really, really sick. And he just is at home and you don't see him. And people are missing from their lives. And so this is very similar to long COVID, and people can't do what they want to do in their normal lives. And so what are the problems been in ME/CFS? And I thought I just might go through this. So clinical diagnosis has been really difficult. It's quite subjective. There are no tests for this condition. And all of the treatment trials that we've had so far have been endpoint, not really very subjective questionnaires and fatigue scores. And it's caused lots of problems. You're probably aware of the PACE trial and the changes to NICE guidelines in this condition. And so there's lots of distrust between patient groups and the medical profession, and it goes both ways. And there's been very little research funding. We've existed on charity funds in the UK, so £50,000 is probably your maximum you can get from the ME Association. So it's a challenge to keep going. It's better in the US. There's some big centre grants now, £20 million. So they've seen the light and they're moving forwards. And what we need to do in the long COVID space is to get proper funding in the future. So what are the symptoms? And these have probably resonate with a lot of people. We've seen these throughout the meeting. So this inability to function normal activities can be quite profound, can be the fact that people can't go into the bathroom, can be too much for some people, but there can be a wide range here. Unrefreshing sleep, we heard yesterday that also is an issue. Cognitive impairment, and POTS and orthostatic intolerance, just some nice presentations in the earlier session. The key one really that discriminates this from, say, depression is the one in the box, so the post-exertional malaise. So the worst things of symptoms after physical effort can be cognitive or emotional. But this is really hard to quantify. We just ask the question, do you suffer from post-exertional malaise? And we need a way of quantifying this, but in a way that doesn't really impact the patients. So these are the 5 key symptoms. There's a whole lot of other things that can be associated with as well. And why are we studying this condition? Because it's pretty horrible to have it. I think that would be the bottom line. So this is a SF36 comparison, looking at physical components and mental components. And it's one of the worst conditions you can have in terms of physical and mental impacts on the patient, worse than things like osteoarthritis, back pain, and cancer. So it's a clear need here. And in terms of how it affects people, there's probably more like 20 million. And unfortunately, with the long COVID and the COVID pandemic, people that now move into this ME CFS class is going to be increased. So the cost to not just economy, but the families is going to be enormous. In ME CFS, around about 85-95% of patients have never had a diagnosis. It's not a proper one because we can't diagnose it well. And again, like yesterday, there was some evidence in long COVID that females are more affected, like they are in many of these autoimmune conditions. So that's a sort of bit of a summary, really. This is a nice slide from Ros Ballings, who's an expert in ME/CFS from New Zealand, and it's more of a sort of potential what could be going on. So this is Epstein-Barr virus, and many of us have this as a child. I had it as a child, and you can be quite mildly affected or more severe. And after a month, most people are back to where they were, so 90%. And then some people will go into this post-viral phase, probably 9% of patients. And if you get stressed or you push yourself too hard, you can relapse. And that's quite a potential problem. But then most people get through that. So you're left with around about 1% that have an ME/CFS type condition. And they may get better or they may stay there. So COVID and long COVID-19 fits quite nicely into this profile, really. And whether we're going to see more cases of ME/CFS is still to be seen, really. So that's an interesting demographic. And what I thought I'd show you, there's lots of work around viruses now, particularly in multiple sclerosis. And so where do the mitochondria fit into a condition where we have studied it in a lot more detail? And there's a recent paper came out in Science in the last month or two, showing quite clearly, we've known for a while that EBV infection, if you don't have EBV infection, you have much less chance of developing multiple sclerosis. And they showed in this paper that there was a clear correlation of EBV as a risk factor. So that's something that's quite new. And it affects B cells, and that's something that will be a theme of the talk as we go through. So MS is a demyelination of the nerves, and you can see that on that slide. And the other thing you see in MS, which is really interesting, is that you see high levels of mitochondria in these myelinated neurons in the patients, much more than you'd expect to see. And what we know is that if you damage demyelinated neurons in a rat, you see recruitment of mitochondria to the site of damage. So the current thinking is that maybe there's more mitochondria because they've been recruited to the site of damage. But there could be other things going on here as well. And I'll come on to that in a minute. So increased mitochondria in these axons when they're in these patients, but could be related to damage, trying to do some sort of repair process. And so this is a sort of where we are with ME/CFS, and I'm going to talk really just about the ones on the left-hand side around the mitochondrial energetic dysfunction. But we do know lots of bits of the puzzle now. We're getting a lot more studies that can be reproducible. So there are things on this slide that we're pretty hard data is coming through. So it's just connecting up the pattern really is there. So in terms of energy metabolism and ME/CFS, what do we know? And one of the first studies only came out 5-6 years ago from a Norwegian group, and they looked at metabolomics profiling of patients and found some interesting data around low levels of amino acids and lipids, and also some interesting research in PBMCs looking at maybe an impairment of pyruvate dehydrogenase activity. This has not been repeated by any group. But the thing that has been repeated, and we started to look at this, is that if you take plasma from patients or serum and you add it to control cells in culture, you do see an increase in mitochondrial respiration. And they show this in the paper, and this is some work from our group on the right-hand side. So when we add patient plasma to human muscle cells with an intracellular oxygen probe, we see an increase in respiration, i.e. a drop in the oxygen in the cells. And this is something that we're currently working on. So there's something going on with the plasma that can impact energetics, and that's research that's going on in other places. And this slide really summarizes the work in PBMCs. Most of the work in ME/CFS has been on peripheral blood mononuclear cells, because that's the tissue we can easily get. And there's a growing evidence there is a problem with energetics. So the top left-hand side is a paper by the group from New Zealand, so proteomic analysis. And the two papers on the right-hand side were by an excellent group from Newcastle, led by Julie Newton. And unfortunately, this group is now disbanded because of funding, because you can't keep going on these small money. And so Karl Thomas produced some really nice bits of work. And on the bottom left-hand corner, there's an interesting study from a group in Australia, and I'll just talk about those briefly. So what's interesting on this slide, so the group by Mr. Ledes basically found there was a complex 5 issue in the patients. So I don't know if I can use my pointer on this. Let's just try. I think so. So what you have here, these are the lymphocytes from blood, and if anyone's ever tried to measure mitochondrial function, it's really difficult to do it in lymphocytes. So the levels are really low. They didn't see significance here, unlike some of the other studies, but there was a tendency for the patients to have low oxygen consumption rates in lymphocytes. But what you can do is you can transform these, and with EVV again, which is potentially really relevant. So a viral transformation will give you a much higher rate of mitochondrial respiration, but that doesn't then show a difference between the two groups. So the oxygen consumption rates are very similar. But if you look at the respiratory chain components of the different cell types, that the ME patients have a huge upregulation of mitochondrial respiratory chain protein, which is really interesting. So something on these cells going into culture with a transformation was able to see big differences between the patients and controls. And then the paper by Karl Thomas, again, taking cells from a patient, so these are myoblasts, these are stem cells derived that would have generated muscle, put them into culture, and then you look at their ability to drive respiration. So the glucose-driven respiration was really low, whereas the galactose glutamine driven respiration was quite normal. So this is showing a couple of things. So you can actually transfer this energetic dysfunction type properties by taking cells from patients and growing them in culture. So this makes you wonder, well, how can you do that? And potentially the genetic, epigenetic pathways that could be imprinted in the cells that could be taken through into the culture, or maybe there's something else that's going into the culture, and that's something that we're quite, so could there be a pathogen that's going through, maybe a virus, maybe a bacteria that's going into the culture with them. So this is some data on just a brief snapshot. We've done a lot of work with metabolomics over the years, and we see differences in patients compared to controls, and this is just showing some amino acid ones that are quite relevant to energetics. So you can see here, this is the relative fold change compared to the controls, and things like glutamine is about half in many of the patients. So these are all down. So there's low levels of amino acids in the patients. And what does this mean? So glutamine is a really abundant amino acid in blood, and it's down by 50%. And so in terms of the glutamine, the blood concentration, 0.41 millimolar. So it's really very, very, very high. So what's going on here? And so one area could be that the patients are struggling with glucose, as was alluded to by the Norwegian group, and we're using other fuels to compensate. So there could be a drive to use glutamine to go through to make ATP, but glutamine is made mainly by muscles, so there could be a problem with the production of glutamine. And so this is an interesting area that we're exploring. So if there was a problem with glucose metabolism going into the mitochondria, maybe by PDH, there's a hypoxic regulation here, which could be also playing a role. So this is an interesting clue. The key thing to mention is we don't see this with all the patient groups, and we're trying to reproduce this. So seriously, all people aren't showing the same effect. So this may be the milder patients that are showing this interesting phenotype. So what do we do next? We've studied PBMCs. What type of things should we be doing? if we have the funding. So I think the time is right to start trying to do some imaging work, to use MRI, spectroscopy, but this is expensive, and this is something we probably need to do. And a key thing is to do multiple measures. We do get a lot of variation between patient controls, and when you start digging deeper into some of these other omics technologies, you do start to see huge variability, even in the control population. So having a multiple measures, but that costs money. You need to do more than one scan on a patient. Using an intervention, either exercise or a cognitive test, could really start to tease out what is changing in these individuals in terms of their metabolism. And that's something we need to do in a sort of safe way for the patients. And there are new things that we could use. Maybe Betty Rahman will talk more about this type of thing this afternoon in her talk, but this dynamic nuclear polarization allows you to look at, say, pyruvate metabolism, so to really tease out what's going on in terms of these metabolites. And I'm going to switch tack a bit now, because I've talked a bit about the ATP. Mitochondria is the powerhouse of the cell, but we now know they do other things. And so for the last part of the talk, I want to talk a bit about how mitochondria might be involved in infection and how that might be of relevance in this condition. So on the right-hand side, you have a JC1 stained neuron, and you can see the brighter regions are where the mitochondria are. And this is a bit of a teaser slide. I haven't got time to go into it today, but a collaborator of ours is finding wall-less pathogens in patients with chronic illnesses, including ME/CFS. You can see here a white blood cell. A little green speck is an L-form bacteria. These are very non-immunogenic, and they can hang out in cells and hide in cells, and you can also culture them. So that was just a teaser, really. And as I said, I've worked on mitochondria for many years. And this is the classic mitochondrial structure that you probably were taught. And these are primary hepatocytes from human liver. And they're very punctate, look a bit like this. Whereas fibroblasts are really different. They're very long, filamentous. And I've always wondered, what's going on here. And this, so this image at the bottom here is from INS1 cell. So these were tagged with GFP in the mitochondria, and then we've got a functional stain staining the mitochondria orange, and you can see there's some cells that are green and some that have more sort of orange speckles, so these are more active, and so they're different even in a homogeneous culture. And so at the time we didn't really think much about this, but there's new work coming through that suggests this could be really important. So this is a comparison of two types of pathways. So if you have a pathogen affecting a cell, you have this PAM model here on the left-hand side, and these molecular patterns you see. And then when mitochondrial dysfunction occurs in disease or in aging, you also see something very similar. But what's quite striking is that the end point of this is the same. And so this innate immune activation can either happen from a bacteria infecting a system or mitochondrial failure. And these two systems could be quite related and quite important. And this is some recent data come from Lena Pernis group at the Max Planck, showing that bacteria like this, Toxoplasma gondii, which is found in over a third of all humans, can cause some quite catastrophic changes to the mitochondrial biology. So in the panel in the middle, you can see that what the bacteria does, it strips off the outer membrane of the mitochondria, and it renders those mitochondria less good at mounting a defense. And so there's another paper from Lena you can read in Cell Metabolism that talks about how mitochondria mount this defense type system. And on the right-hand side, you can see the red bacteria surrounded by the mitochondria in green, and it's as if they're being engulfed by the mitochondria, and that mitochondria is probably trying to control the replication of those organisms in the cell, and maybe trying to get rid of them. And so this is something that's quite new, and obviously a growing area that could be highly relevant here. I wanted to mention this. So this is some research from an Italian group led by Alessandro Morelli. They've been working in this area for about 15 years. Their research hasn't been widely accepted by the community, but I think it's really potentially really important. So this slide is just showing you, if you look at the mitochondrial area density in different tissues, they are really different. And you can see from this that the brain has only 5% volume of mitochondria, whereas it's one of the biggest users of oxygen in the body, the highest user of glucose. So how is it making its energy if it has so few mitochondria? And the other cell of interest is, again, is this B cell, the plasmablast. These are the cells that make the energy, the antibodies producing cells. And again, when they go into this highly differentiated state, you find lots of ER structures forming, but very few mitochondria. And if you look at the price paper shown below, there's a huge cranking up of the TCA cycle enzymes and respiratory chain enzymes. Well, where are they? They're not in mitochondria. So what Alessandro and the group are saying is that a lot of aerobic ATP synthesis occurs in membrane structures in humans and not in mitochondria. And this hasn't gone down very well with the mitochondrial community, but I think they need to start listening to what they're saying. Because if you go back and look at this slide, myelinated neurons don't have very many mitochondria compared to an unmyelinated neuron. So maybe that makes them vulnerable. And it's the myelin around them that may be the thing that's driving the ATP production. And a beta cell, again, why beta cells? Maybe they're more vulnerable to EBV infection, and that's part of the process. So this is something we need to bear in mind. And this is some nice work by Professor Prosti from Germany. He's a virologist, and you can see on the left-hand side what happens when chlamydia affect cells, and again you get this similar network structure that I showed you before with the toxoplasmosis, as if the mitochondria are engulfing the bacteria. And whereas a virus does something completely different. So if you activate herpes 6 virus, so the red cells have had it reactivated from the latent form, the mitochondria become really punctate. So you can imagine a scenario where you don't just have a bacteria in a cell, you may have a virus as well. And what happens when you see the two? And so certain tissues like the heart and the liver have a lot of mitochondria. So maybe they're very good at dealing with these viruses, but the brain seems to be lacking. So obviously there's a blood-brain barrier that is protective, but what if things get in there and maybe there's a problem with the brain in dealing with viruses, etc. And so if there are pathogens, where are they? You know, what's the evidence that there's levels of infection? And there really isn't good evidence for a full-blown infection in patients with ME/CFS when they suffer from the disease. And it's the same for long COVID. I don't think there's probably much virus left. So what could be going on? And so this is an interesting systematic review looking at diseases, schizophrenia, depression, mental health, and chronic fatigue. And what you find is that there's a lot of evidence of a permeable gut. So you find high levels of LPS in the blood, you find other markers. So the SCD14 is a liver marker linked to the presence of pathogens, and the zonin 1 is a marker of gut integrity. It's tight junction protein. So there are evidence of pathogens in the bloodstream of these people, but at low levels. So they're getting in there, but what are they doing, and how can we think about that as a mechanism? And a recent study from one of the centers in the NIH funding centers in the US, from Lipkin's group, has shown quite nicely in a recent PLOS paper that there are things going on again with the B cell expansion. So maybe there's a couple of things that are driving this antigens, a couple of antigens. And also something hidden in the paper was this LL37, which is an antimicrobial peptide, which are raised quite high in these patients. And also these microbial peptides back in the 70s, they also have an anti-mitochondrial effect. So there's stuff emerging here around, there could be something going on. And just the sort of final slides really. So this is some data I thought I'd show you from Soft Cell Biologicals in Utah. So they've been doing this work for about 5 or 6 years, 2,000 subjects. They've been looking at autoimmune conditions. So this is the data from the ME/CFS and the fibromyalgia groups. And what I'm showing here is they've found a way of culturing these organisms in the lab from blood. And what they're finding is there's much, much higher levels of culturable organisms in these conditions. So the ME/CFS and the fibro, much more than the autoimmune conditions, which also are high. As you get older, we have more culturable organisms. So this might be a factor of just getting older. And these organisms, as I mentioned, are really non-immunogenic. They sort of hide away, you know, in our cells and tissues. And if they're in the brain hiding away, then they could cause trouble. And we're also finding them in tumors now and around in cancer. So this is a new and emerging area which could be really relevant in chronic conditions. And just to finish off really, so this is an idea I've had. So, you know, I've always wondered, how can mitochondria do all these things? You know, they make energy, they make cofactors, they do reductive carboxylation, and maybe they're now responding to pathogens. So can one mitochondria do all of those, or do we have them compartmentalized? So do you have in a cell, some mitochondria make energy, cofactors, others make building blocks, and then a pathogen? And there's this balance that's important. And so if you have a healthy muscle cell on the top right-hand side with a little bit of an infection, you can cope, you have a good mitochondrial energy producing system. But if that cell becomes compromised with you know, the pathogens, you overdo it, you allow the pathogens to take over, and then maybe the balance is wrong, then you have less ability to make energy. And if you think about a brain cell with even less mitochondria, it's going to be even more compromised. And so just to summarize really, what do we know about energy dysfunction and where does it sit, which is a key question in the disease process. Is it a driver, or is it something that's a consequence? So there's clearly an evidence for an energetic defect in PBMCs from patients, and that's been established in many labs now. It seems to be more of a problem of flux as fuel rather than mitochondrial damage. The mitochondria seem to be fairly well and fairly intact. Reduced levels of amino acids in blood, in lipids, that's been seen by quite a lot of groups now. So there's something going on at the metabolic level, but what it means is another question. Maybe it's increased fuel use or something else. There is a plasma factor, but we're starting to see it in controls as well. So we're not quite sure what that means. So there's clearly something in the blood of anybody that can change mitochondrial respiration rates, and we're just working on that. We don't really know much about what's going on in affected tissues, and this needs to change. We need to do more on muscle and brain. There's work going on in Oxford that's looking at this, but we need more of a budget. We can't work with £50,000 from a charity to do this. And pathogens, it's an area that we're very interested in. How do we study them? We can do deep sequencing now. We can pick them up in blood. We can culture them. So there's more we can do. And just to finish there, this is what we'd like to see. Really understand the system. What are we doing? We're really focusing on the gut as a potential driver, and we're bringing in these other areas to try and work with that. I've got 2 PhD students that just started, so we're okay for the next couple of years. So we can do something which is quite nice for us working in this space. Where these fit in is another question. Obviously, we can't do everything, but they fit in somewhere. It's been mentioned already in this meeting. So just to acknowledge my group. So I've got a really good group in the lab. So Jamie, who was on the previous slide, who works on the bioinformatics, and at the bottom there's ME Association. And actually the local patient groups have been really helpful in terms of funding, and we work with a couple of groups here. So one last plug, we have a meeting in April called Mitox. This is probably the best one-day mitochondrial meeting in the world. You can ask Andrew Murray, he comes every year. So I will stop sharing and sign off if I can work out how to get out of my slides.
Thank you very much, Karl. I'm sure that this will generate some discussion points in the panel discussion.